So we're still in chapter 22. We just kind of started into 22 on a lot of the anatomy of the respiratory system. And we left off here talking about, we've made our way through a lot of what we call the conducting zone structures. So basically from the nose, down the pharynx, into that larynx, our voice box, we talked about that. We talked a lot about the trachea and those hyaline cartilage. I call them rings, but they're really these C-shaped structures. So we left off here. Once that trachea branches behind the heart, it branches into a right and a left. We call those the bronchi. So we're going to walk through the bronchi and the subdivisions. Now those bronchi, and you can actually see that right here. So the trachea would come down and you can see where it branches right here. This is happening right behind the heart. It's going to undergo about 23 orders of branching. So it's going to keep branching and the branches will get smaller and smaller and smaller. And this is called the bronchial tree. And so I'm going to talk a little bit about this bronchial tree in a minute, but I want you to notice all of the colors on this bronchial tree because those are actually really important. We'll come back to that in a few minutes. So where the trachea first branches are into what are called primary bronchi. So remember the term bronchi is plural, bronchus is singular. So this is plural. And there's a right and a left primary bronchus. And each of those are going to enter into sort of a divoted area in the lungs that we call the hilum of the lung. Now, if you look at this image, you can tell that right primary bronchus is a little bit wider. So remember, here's the right side. Okay, so look at that primary bronchus. It's a little bit wider. It's a little bit shorter. And it's a little more vertical than the left primary bronchus. So if somebody aspirates food, if it makes it into their trachea and it gets lodged, a lot of times, if it moves down the trachea, where it's going to get lodged is over here in the right primary bronchus because it's fatter and it's more vertical. So that is a much more common place for objects to get lodged. Now, each of those primary bronchi are going to branch again into what are called lobar bronchi. And these are named because these are going into the lobes of the lungs. Okay. Now, remember, in terms of your lungs, if we look at the right side has 3 lobes to the lungs, one here, and one down here. The left side only has 2 lobes, one here and one here. And the reason for that is because the heart sits a little left of center, so it's taking up space on that left side. So you really only have space for 2 lobes. And so when it comes to lobar bronchi, you're going to have, they're feeding the lobes. So you'll have 3 lobar bronchi on the right and only 2 lobar bronchi on the left. And now each of those lobar bronchi are going to branch again into what are called segmental bronchi. Now, a lot of times we'll also call these bronchopulmonary segments. Means the same thing. There are 10 segments in the right lung because there's more lobes, it's bigger. And there's usually only 8 segments on the left lung. It varies from person to person. Remember, there's anatomic variability. If someone has a problem in their lungs, if the lung is diseased, it used to be that they would remove the whole lung. Then it became, well, we can just remove one lobe of the lung. And now we know that we can remove just one of those bronchopulmonary segments. So that's about as small as they can get in terms of removal if they have to. So let's come back to this image down here. Remember, I told you it's color-coded for a reason. The color coding here is showing you those 10 different bronchopulmonary segments on the right and the 8 different bronchopulmonary segments on the left. So that's why it's color-coded. Now, those segmental bronchi are going to continue to branch and divide, and those branches are going to get smaller and smaller and smaller. And they're going to turn into what are called bronchioles. Now, remember when we did vessels, I told you a lot about how anytime it's eol at the end, like arteriole, veinole, bronchiole, it means it is microscopic. It is tiny, okay? So bronchioles are less than 1 millimeter in diameter. Terminal bronchioles are the smallest. These are less than about 0.5 millimeter in diameter. So these are tiny. And so what we're going to see is going from the primary bronchi, right where that trachea branches, as we move to lobar bronchi, segmental bronchi, all the way out into bronchioles, we're going to see a lot of structural changes happening. One thing we're going to see is that the cartilage rings that we saw within the trachea, we see these in primary bronchi, these are going to become absent in the bronchioles. So once we get down into those really, really tiny bronchioles, we no longer have those cartilage rings holding them open. The epithelium, so the lining is going to change. So remember, the lining way up in the larynx, the lining is still a stratified epithelium, and it's there for protection because sometimes food goes down the wrong pipe. As you move down the trachea and out into those bronchi, it changes into a pseudostratified epithelium with a whole lot of goblet cells, right, making mucus. Well, it's going to change as those bronchi branch into bronchioles, and we're going to see the cilia and the goblet cells become much less sparse as we get down deep into the lungs. And then also the amount of smooth muscle is going to increase. So you're going to see more and more smooth muscle as we get into those bronchioles. And this is important because it's going to allow those bronchioles to dilate and get big if you need to get more oxygen in, or they're going to constrict and get small. Okay, so that smooth muscle is important as we move out into those bronchioles. So everything that we have walked through so far in chapter 22 has been conducting zone structures. Remember, conducting zone means we are conducting the air all the way down deep into our lungs. Now we're going to move into what are called respiratory zone structures. And these are really tiny structures. These are going to include the respiratory bronchioles, the alveolar ducts, and the alveolar sacs. So the respiratory bronchioles are going to feed in to alveolar ducts, which are then going to dump into what's called an alveolar sac. An alveolar sac, you're going to look at these in the lab. They look like a big bunch of grapes. Okay, that's an alveolar sac. Looks like a big bunch of grapes. And one of those little grapes within the sac is called an alveolus. And you have within your lungs about 300 million alveoli making up your lungs. Now, if we were to lay those out, it would be the surface area of a tennis court in your lungs. So think of the size of a tennis court. That is the surface area you have for gas exchange in your lungs because of all these tiny little alveoli. And so again, the job of your alveoli is we are going to conduct the oxygen all the way down into those little grapes, those alveoli. And once it gets there, this is the site for gas exchange. This is where we're going to take the oxygen, move it across the alveoli wall, and put it in our bloodstream. Okay, so the site of gas exchange. This is a great picture because you can see in this photo, especially the one on the left, you can see the primary bronchus. You can see how it branches into lobar bronchi. Then you can see all those different segmental bronchi. You can see it's coming all the way out here into bronchioles. And then they blow that up really big. So these are the respiratory bronchioles. They're tiny. And you can see they're feeding into this alveolar sac. And like I told you, it looks like a big bunch of grapes. One little grape is an alveolus. And so if we look at this under the microscope, and you'll see some images of this in the lab, we can see here the respiratory bronchiole. So you can see that's where the air is coming through. And you can see it's ending within those alveoli. And so you can see all these little alveoli in here, those thin-walled alveoli. So really what I want to spend the next couple minutes talking you through is, since your alveoli, that grape, is a site of gas exchange. So I told you when you bring that oxygen in, you take it into that alveolus. Now you've got to move oxygen across the wall of the alveolus and into your bloodstream. The wall of the alveolus and the wall of your capillary, that is called the respiratory membrane. This is what you have to move the oxygen across. So it's basically the wall of the alveolus and the wall of the vessel, the capillary, and their basement membranes. Now, for gas exchange to be effective, the respiratory membrane has to be really thin. The thicker it is, the harder it's going to be to pass those gases back and forth. So it is a really thin membrane. It is registered about half a micrometer in thickness. And gas exchange happens just through diffusion. So remember the term diffusion. It means going from a high concentration where you have a lot of something to a low concentration where you don't have much of something. That's all diffusion means. So just think about oxygen. When you breathe oxygen in, it goes all the way into your alveoli, okay, where you have a lot of it. And remember, we have pumped oxygen poor blood from our heart out to our lungs. That's that pulmonary circuit. So now in our blood, we don't have much oxygen. So oxygen's just going to move through diffusion. It's going to go from your alveoli, where you have a lot of it, across the respiratory membrane and into your bloodstream, where you don't have much. Okay, and that's how we're going to fill up our blood with oxygen. Now, I have been mentioning oxygen this whole time, but in reality, it's gas exchange that's happening in the alveolus. So let's also talk a little bit about CO2. CO2 is a waste. We have to get rid of it. This is also going to move through simple diffusion. Remember, when your heart pumps that blood through the pulmonary circuit, the blood going to your lungs is blue. It's spent blood, has no oxygen in it, but it's filled with CO2, has a lot of waste, and we got to get rid of it. So when the oxygen's moving into the blood, the CO2 is moving out of the blood. It's going from the bloodstream across the respiratory membrane into the alveolus. And when you blow out that CO2. So you breathe in oxygen, put it in your bloodstream. From your bloodstream, you're going to take that CO2, put it back in your alveoli and blow that out. Okay. So CO2 is moving from your blood into your alveolus and then out of your body through exhalation. Whoops. I'm just trying to write exhale up here. So this image is trying to combine the alveoli and that alveolar sac, that big bunch of grapes, with the pulmonary capillaries. So all those little capillaries that are around the lungs. Again, this is the pulmonary circuit. So your heart has pumped this oxygen poor blood to your lungs, right? And you can see that it's breaking off into these little capillaries that surround those alveoli. And again, remember, the respiratory membrane is what we have to exchange gases across from the wall of the alveoli through the wall of that vessel and into the bloodstream. And that's how we're going to fill up the blood with oxygen, and we're going to send it back to the heart through those pulmonary veins. Okay. Now, this is an image. This is a scanning electron micrograph where they have taken out all of the alveoli. And the only thing you're looking at here is just the pulmonary capillaries. So you can see how rich in capillaries the lungs truly are. There's tons of capillaries in the lungs surrounding those alveoli. And I think you can see that really well from this image as well. So you can see all that great blood supply, because the whole point is gas exchange between the lungs and the blood. Now, I want to talk to you a little bit about the alveoli. We're going to talk a little bit about the cells in the alveoli and what is making up that respiratory membrane. So most of the cells in your alveoli are called type 1 cells. These are the cells that are responsible for gas exchange. So it makes sense that this is going to make up the majority of the cells in the alveoli. So let me show you a picture. Okay. You are looking at, like here, for example, I'll circle it. This is 1 alveolus. Okay. And if you look at this image, all of these cream colored or tan colored cells, those are type 1 cells. Right? They're making up most of the alveolus. Gases will move back and forth across those cells. Another kind of cell is something called a type 2 cell. These are scattered throughout the alveoli, and their job is to make surfactant. Okay, so let's look at this picture. The type 2 cells are these green ones here. So you can see they're kind of scattered around those alveoli. And the job is to make surfactant. Does anybody know what surfactant is? No? You use it every day. It's in soap, it's in shampoo. Surfactant is a molecule that cuts the surface tension of water. You find it in detergent, laundry detergent. And the reason that's important is you want to cut the surface tension so that soaps can work better, right? Well, why on earth would you ever need surfactant in your alveoli of your lungs? Why would you need to cut the surface tension of water in the lungs? The reason is your alveolus is coated with water. It has a nice thin coating of water in it. There's moisture in there. It's keeping it moist. This is a moist membrane. Problem is, water has a really high surface tension. If you ever see like water striders on the top of a pond, they are sitting on top of that water because of surface tension on the water. Have you ever done the penny experiment where you take a penny and you put drops of water on it? Has anybody ever done that? How many drops of water can you put on a penny? It's a lot, right? It's like 70 drops of water. Like you start and you're like, oh no, I can do like 5 drops. And then you keep dropping and the mound of water will bubble up on that penny. It's amazing. Water has a really high surface tension. And what that means is that water molecules are attracted to each other. So if inside this alveolus, you have a coating of water, the water molecules are so attracted to each other because of that surface tension that they pull the alveolus closed. We don't want that. So the reason why you have cells, these type 2 cells, is they're releasing surfactant and they're cutting the surface tension of that water so that your alveoli don't get pulled closed by that tight, high surface tension. I'm going to talk about this later, but I might as well mention it now. Babies, premature babies, when babies are born early, have you ever heard where if they're born early, the doctors might say, oh, we want to keep that baby cooking longer because the lungs aren't fully formed. Have you heard that? All of the organs are completely formed by the time that baby is by the time a mom is in her 2nd trimester. So at 16 weeks, for sure, those organs are all formed. So what we really mean is that if a woman delivers a baby before 38 weeks, it's not that the lungs of the baby aren't developed. It's that these type 2 cells aren't making surfactant yet. So a lot of times when babies are born early, before 38 weeks, they get put in the NICU and they have to be given synthetic surfactant so that their lungs don't collapse, so that their alveoli don't collapse until their own type 2 cells start making surfactant. Okay. When a baby is born, the first thing they have to do is take a breath, right? That's the first thing they have to do. And when that baby takes a breath, it's going to be the hardest thing it does because it has to inflate the entire bronchial tree and all the alveoli. So that first breath takes a lot of effort. So you can imagine a baby that's born too early that doesn't have surfactant, every breath is like the first breath. Because after every breath, when they blow back out, those alveoli collapse. And they have to do that all over again. They've got to take their first breath and inflate everything again. And so this is why babies that are born too early are in the NICU. They're given synthetic surfactant until their own type 2 cells can start making it. The other kind of cells that we find deep in the alveoli are alveolar macrophages. Now, you have heard of macrophages before. We talked about these in chapter 21 when we talked about the immune system. Another term for macrophages are dust cells. Now, a lot of times we find macrophages that move throughout loose tissue, like they move through your areolar tissue and your dermis. And their job is to just kind of patrol the area and clean up anything that shouldn't be there. But we also have macrophages that are permanent residents in certain organs. For example, you have some that live always in the liver. They don't ever leave the liver. You also have some in the lungs, and they never leave the lungs. And their job is they're like the janitors for your lungs. They are the lung housekeepers. They move around the alveoli and they clean the alveoli. They get rid of debris. Think of them as like little trash cans, and they wander around, and when they see dust or debris, they pick it up and put it in the trash can. And that's what they do. And then once that macrophage, once that trash can is full, it jumps on the mucus escalator and it gets pushed all the way up the trachea. You swallow, it comes out the other end. So this is how you are constantly cleaning out your lungs and your alveoli. You will carry about 2 million dead macrophages per hour up your cilia and into your throat and swallow them. So again, remember, we talked about how people who smoke, how they paralyze their cilia for 1 hour. One cigarette will paralyze cilia for 1 hour. So that means that all the dirty mucus in the upper respiratory tract sits around, but that also means that those macrophages, your lung janitors deep in your alveoli, they can't hop on the escalator because it's not running. So they sit in the lungs. You've ever seen pictures of a smoker's lungs? What do they look like? They're black, right? That's because of all of that buildup in the lungs that can't be removed. Now, I've had people ask before, so what happens then when someone stops smoking? Let's say they're a heavy smoker and then they stop smoking. Their lungs can clean out. Once that mucus escalator starts working, those macrophages will start putting stuff in trash cans. Problem is they've got a lot of backlog to do, right? So it takes a long time to clear those lungs out, but they can get clear. Somebody asked me about vaping. So I looked that up, and I can't remember the name of the chemicals, but it says that vaping, specifically, there are 2 chemicals that are linked to the flavoring that's in vaping, the vaping cigarettes, that are never meant to go through the respiratory tract that paralyze the cilia. So there's a study done in Harvard in 2019. So I found that, and there are 2 very specific chemicals that are in 90% of the vaping cartridges that are on the market. So it does the same thing that someone who smokes a cigarette. So before we go to the next slide, I want to look at this picture over here on the right, because this is showing you the respiratory membrane. So remember, when you breathe in and the oxygen goes deep into your alveoli, you've got to move it into your bloodstream. So you've got to move it across the alveolar wall, across the capillary wall, and into your bloodstream. And that's our respiratory membrane. And so you can see here, that is the respiratory membrane. So it's the alveolar wall and the capillary wall, and they're fused basement membranes. Now, I already mentioned the type 2 cells that help to produce that surfactant to cut down on the surface tension of water. Now, in terms of alveoli, your air sacs have a lot of elastic fibers in them. This allows them to expand and contract. They can stretch when you take big breaths in, and then they can recoil. So they're nice and stretchy because of these elastic fibers in there. But also your alveoli have alveolar pores. What's a pore? It's just a hole, right? So your alveoli have all of these holes in them that connect them to their neighboring alveolus. Okay, so they're all connected together. And this just makes sure that the air pressure as it moves into your lungs and into your alveoli is equal between all your alveoli. It's spread out through all of those alveoli. So you can see from this image, you can see all of these pores here. So they're just connecting all those alveoli together so that the air can move equally throughout the entire lung. Now, I want to mention, I know we haven't really talked about the lungs in general, so that's kind of the last thing we're going to talk about before we get into the mechanics of breathing. Let's talk a little bit about the lungs, what they look like, where we find them. So remember, they're found in the thoracic cavity, which is in the ventral body cavity. The root is basically the part of the lung where you have the blood supply and the bronchioles going into and out of the lungs. The costal surface of the lungs is basically the part of the lung that would be next to the ribs. So the costal surface is really in the front of the lungs, on the side of the lungs, and on the back of the lungs. So it's pretty much all over everywhere else. So the root is going to be kind of the middle part of the lung that is right next to the heart. It's next to the trachea, next to the mediastinum. And then the costal surface is everything else where it's next to the ribs. Again, your lungs are basically alveoli, little air sacs. Our preserved specimens, because they are preserved, the tissue doesn't feel like fresh tissue. It changes the texture when you preserve it. Does anybody hunt in here? No? So nobody's ever like field dressed a deer? No. If you are a hunter, if you've ever field dressed a deer and you have touched fresh lung tissue that's not preserved, it is really squishy. It almost feels like a really delicate sponge because of all those alveoli in there. So this image is kind of showing you the surfaces of the lungs. So again, right here, this part in the middle right there where it's connected to the mediastinum, if we go back to this image, that's the root. Okay, and now if we look at everything else, so everything else back here, around the sides and in the front, that would all be the costal surface. Okay, so that's just tying those two terms to an image. Now, some of the other parts and pieces of our lungs, the apex of the lungs. Remember, the apex is always the pointy part of an organ. So think back to the heart. The apex of the heart is at the bottom. It's where the heart's kind of pointy. Now, the apex of the lungs is at the top of the lungs. This is where they are more pointy. Okay, so let's look at a picture. Okay, so you can see the apex of the lung here at the top, where it has more of that point to it. So just under the clavicle. The base of the lung is where it's more flat. This is where it is basically sitting on top of the diaphragm. So let's look at a picture of that. This would be the base of the lung down here, sitting on top of that diaphragm. The hilum. The hilum is basically an indentation within the lungs on the towards the mediastinum. And it is where the blood vessels, the bronchi, the lymphatic vessels, nerves, where they're entering and exiting those lungs. So the hilum has sort of an indent. And so we can't really see it on here. The hilum of the lung would be in here. Okay. Kind of deep to that image. Now, I mentioned to you that the left lung is smaller than the right because of the heart. The heart sits left of center. The left lung has 2 lobes. The right lung has 3 lobes. The left lung has a notch cut out called the cardiac notch. And this truly is a notch cut out for the heart. That's why we call it the cardiac notch. Now, if we look at this image, not this image, but let's go back to this one. If you look at the image, you can see that cardiac notch here on the left side. So you can see a little notch cut out for the heart. And again, remember, our lungs are separated into these lobes. We have superior and inferior lobes, and they're separated by fissures. So you can see, for example, the superior lobe of the left lung, the inferior lobe, and it's separated by, those are separated by that oblique fissure. Now we can look at our lung and break it into the smallest subdivisions visible to the naked eye. These are called lobules. So if we look at this image, you can see the lobules kind of within this lung. It's basically these little, I don't know, they look like little lobules. I don't know how else to explain it. The little lobules, like here is one and here is one. Those are the smallest divisions that we can see. And these are served by bronchioles and their branches. They kind of give the lung its texture. Now, when we look at our lung, it is supplied by two circulations. When we look at the blood supply, there's the pulmonary circuit, and then there's also the bronchial circuit. So let's walk through that. The pulmonary circulation. Pulmonary circulation is low pressure. There's not a lot of pressure behind that blood, and it's a high volume. There is a whole bunch of blood moving through that pulmonary circuit. So this is the one that we talked about at the beginning of the semester when we did the cardiovascular system, right? The pulmonary circuit. So this is where the right ventricle is pumping out to the lungs through pulmonary arteries. Remember, arteries carry blood away. And then it goes to the lungs, fills up with oxygen, and pulmonary veins bring back to the left side of the heart, and you pump out to your systemic circuit. Now, the pulmonary arteries, remember, these are deoxygenated. So these are blue in color. Remember, artery means away. So remember, the whole point of the pulmonary circuit is to take it out to the lungs and fill it up with oxygen. Pulmonary arteries are going to branch profusely and turn into those pulmonary capillaries that we find around the alveoli. We're going to put oxygen into the bloodstream, and pulmonary veins will bring it back to the heart. And the pulmonary veins, remember, they're going to be red because they are carrying oxygenated blood at that point. So this is pulmonary circuit. Remember, it doesn't follow the typical red and blue rule that we usually see with arteries and veins. It's kind of the opposite. Now, remember, all of your body's blood volume will pass through the lungs about once a minute. Remember, we call that our cardiac output. Your heart will pump out your entire blood volume about every minute, and that is going through the pulmonary circuit and the systemic circuit. Now, that means because so much blood is moving through the pulmonary circuit, the capillary endothelium, that means the lining of the capillaries in the lungs, contains enzymes that can act on substances in the blood. That should make a lot of sense why it's here. It's here because you're moving your entire blood volume through this area every minute. It's guaranteed to go through here every minute. Whereas if you look at your systemic circuit, your whole blood volume moves through the systemic circuit, but that doesn't mean that it's all moving through the splenic artery, right? Some's going through the splenic artery, and some is going through the superior mesenteric artery, and some is going, right? It's going all different places, but we know it's all going up to the lungs. So this is why the endothelial lining of all those capillaries in the lungs will contain enzymes that can work on substances in the blood. Enzymes like angiotensin converting enzyme. This is one that can activate a blood pressure hormone anytime your blood pressure gets off. Now, we also have, other than the pulmonary circuit, we also have the bronchial circuit. And the bronchial circuit has a much higher pressure and much less blood moving through it. The bronchial circuit is basically providing blood to the lungs themselves. So remember how when we talked about the heart, we talked about how your heart pumps the blood everywhere. but we also have to feed the heart muscle itself because it's an organ. This is the same concept. We are taking blood out to our lungs so we can fill it up with oxygen. That's the pulmonary circuit. But your lungs are organs. We have to feed them as well. And that's the bronchial circuit. Now, the reason it's high pressure is because this is oxygen-rich blood that's coming from the aorta. This is a branch off the aorta, so it's under high pressure. And it's going to enter into the lungs at the hilum, and it's basically supplying all of your lung tissue with the exception of the alveoli. Why does it not need to supply the alveoli? They're already getting that oxygen, right? So they don't need to be supplied by these bronchial arteries. Now, one other thing that I want to talk a little bit about are the pleura. So remember, these are our serous membranes that sit around the lungs. So remember, serous membranes, you have them all over the body. You have a serous membrane that sits on the heart. You have some around the lungs. You have some around your abdominal pelvic organs. And serous membranes are double layered. Remember, you have a parietal and a visceral serosa. And when we talk about the serous membranes around the lungs, we call them pleura. So we have a parietal and a visceral pleura. Remember the term parietal? I always remember cavity. This is the one that covers the thoracic wall. It's basically the outermost serous membrane. The visceral pleura, when I see the word visceral, I think organ. This is the one that's sitting on the organ itself. It's sitting on the lungs. So it just sits right outside the lungs. And these are separated by a pleural cavity. So they are separated by a space that's filled with fluid. And the fluid is called pleural fluid. And the whole point of this, let me show you a picture. So we can see, I'll just draw a red line here. This would be the visceral pleura. You can see it's sitting right on top of the lungs. Okay, and you can see it covers the entire surface of that lung. And then let me switch colors. I don't know if I can do that here or not, but we'll try. The blue line would be the parietal pleura. So this is the one that sits farther outside along that cavity wall. Okay, and you can see that they are separated by a space. There's a space between them. And that space is the pleural cavity, and it's filled with pleural fluid. Okay, and again, the whole point of this is that you reduce friction. You don't have a lot of space in your body. There's not a lot of empty space. So anytime you have this little tiny amount of space filled with fluid, what that means is that anytime you have an organ that's really active, like your heart or your lungs, and it's constantly moving, what you don't want is for it to move and rub on a neighboring structure, because that generates friction, and friction generates heat. And heat causes swelling, and swelling causes an immune response and pain. And we don't want any of that. And so by having this little bit of space, this pleural cavity, now every time your lung moves, there's fluid in there, and there's basically no friction being created. Now, I want to talk a little bit about some imbalances that are associated with this. So the first one listed up here is pleurisy. Pleurisy is when those pleural serous membranes become inflamed. They become inflamed, they become swollen. This a lot of times can result from an infection. So if the infection can spread from your lungs into that serous membrane, it can cause swelling. So pneumonia is really, really common at causing something like pleurisy. The problem is when those serous membranes become swollen, now they're not doing their job. Now you're not reducing friction. What's happened is the parietal and the visceral pleura have swollen so much that the pleural cavity is pretty much no longer there. So now every time you breathe in and out, you've got friction. And that results in a lot of pain. So people with pleurisy have pain every time they breathe in and out. This is usually treated with steroids or anti-inflammatories. Take the swelling down. so that somebody doesn't have this pain. And then antibiotics if it's caused by an infection. A pleural effusion. This can happen anytime fluid accumulates in that pleural cavity. So anytime you get too much fluid in that space between the parietal and the visceral pleura. So here's a picture of that. And I mentioned to you a couple minutes ago that actual lung tissue is really soft and squishy. So if you start adding fluid in here, that lung tissue starts getting compressed. And now it makes it real hard to breathe and really hard to move oxygen into your bloodstream. And so this is, if you have extra fluid building up, they have to go in and pull that fluid out. And that is a process called a thoracentesis, a pleural tap. This is where they can go in and remove the fluid, or they can remove air if there's air in there as well. And you can see that here.